Good afternoon. Thanks for joining us for Medical Grand Rounds. I'm David Schwartz, Chair of Medicine. This presentation is going to be recorded for future reference. You'll be muted when you enter the Zoom room. However, at the bottom of your screen is a Q&A icon. Use this icon rather than the chat feature to submit your questions. At the end of the presentations, I'll moderate the question-answer period with the presenters. SARS-CoV-2 is a beta coronavirus that was initially identified in China at the end of last year, and by last account has infected more than 2.5 million people around the world and accounts for 180,000 deaths. While social distancing, testing, and case isolation can reduce the burden of disease, and some therapies may prove to be effective in treating the infection, a vaccine is clearly the most definitive approach to stopping this pandemic. Today, we have three excellent speakers to help us understand SARS-CoV-2 vaccine development. Rosemary Rochford will introduce the topic and discuss approaches to humoral immunity. Ross Kedl will discuss T-cell immunity and approaches to vaccine development. And Florian Krammer will discuss the specific immune response to the virus and vaccine design for SARS-CoV-2. Rosemary, I think you're first up.
Great, and thank you everybody for attending. Today, I'm going to talk about, and I need to get my cursor here, humoral immunity to SARS-CoV-2. And I wanted to focus, since this is obviously a new pathogen, what we can learn from studies on other coronaviruses. So to begin with, I wanted to start with what I call a humoral immunity primer. Just not all of us think about this all the time, and what we remember might be way back in some of the classes we've taken. So when we think about antibody-mediated response, obviously developed by, induced by B cells, and we think about both the primary response and the recall response. During the primary response, we have sort of the first phase where we see the antigen, in this case the virus SARS-CoV-2, interacts with the naive B cells and gives rise to both short-lived plasma cells and germinal center B cells. Now those short-lived plasma cells make an antibody response, typically IgM, not a high affinity and not a long-lasting response as well. The germinal center B cells can then interact with the antigen again in the context of follicular helper T cells in the lymphoid tissues. And that can ultimately give rise then to the long-lived plasma cells and the memory B cells. So that's the primary response. During the recall response, those memory B cells, so we see an antigen, the second response, you see the antigen again. Those memory B cells can differentiate to plasma cells or also re-enter the germinal center to undergo affinity maturation again. So importantly to think about this, and when we're measuring immunity to a pathogen, we're looking at two different things. First of all, we have the memory B cells that when re-stimulated in a recall response, make a rapid response, differentiate to plasma cells and a high affinity antibody on re-encounter. We also have the plasma cells, both those short-lived low affinity ones as part of the phase 1 response, and then we again have the long-lived plasma cells. Those home to the bone marrow, and those are the source of the circulating antibodies that we measure in the peripheral blood when we're trying to look at who's been exposed and infected. Those are not the antibodies that we measure. We think about the memory B cells are the ones like the long-term memory and generate that more rapid response on re-encounter with antigen. So those are the two sources of antibody responses. And we think about the types of antibodies, as I said, IgM are those first responders, not the best, but good markers of a primary infection. IgG is what we measure in the circulation. And IgA, we can detect it in the circulation, but primarily we think about that as a secretory antibody or mucosal antibody response. Now, during a viral infection, we think about two main functions for the antibodies. The first is the neutralization of infection, the ability of the antibody to bind to the virus and neutralize that infection of a cell. The second, we don't talk about quite so much, but it also does play a role, is antibody-dependent cell-mediated cytotoxicity. Shown on the bottom here is a time course of the immune response to a typical acute viral infection. And essentially the virus peaks and rises within the first few weeks of infection. And we then see the emergence of both neutralizing antibodies, CD4 T cells and CD8 T cells specific for that pathogen. Those wane over time, but you can see the time scale here. We're talking in 25 to 75 years. So for the lifetime of the host, neutralizing antibodies are there for a long time, and sort of really should typically for most infections stay high, have a long half-life. When we focus then on coronaviruses, which is obviously the focus of our conversation today, we want to talk about sort of the targets for antibody-mediated neutralization. And shown on the left panel, and this is a nice review that just came out in Trends in Immunology, you can see here's our coronavirus with the spikes that form that crown for the virus, so classic feature of it. And those bind to the cellular receptor and mediate membrane fusion. And once it gets inside the cell, undergoes viral replication and then finally egress and viral release. When we look at targets for neutralization, antibody targets for neutralization, we're focusing primarily on the spike protein that binds to the cellular receptors and can inhibit that entry, and also antibodies that would target membrane fusion as well. So both receptor antibodies that target and block receptor binding, and antibodies that would block membrane fusion. Now we can have natural antibodies, the ones in our body that are made to fight the virus, but also shown here is we can develop antibody-mediated therapies that could then inhibit that binding. So since SARS-CoV-2 is fairly new and we don't have any information on long-lasting immunity to it, what do we know about immunity to the other coronaviruses? And so I'm going to talk about a few studies, one with infection of volunteers with the seasonal circulating coronavirus that causes upper respiratory tract infections, and then studies done with the SARS-CoV-1 and the MERS-CoV. So this study was published in 1990 by the MRC Common Cold Unit, which is a great title for a unit to work in, I have to say. And if you look at this, what they did is they took volunteers and infected 16 volunteers with the seasonal coronavirus, upper respiratory tract infection, and then looked at the development of antibodies over time, specific antibodies to the virus, and then also specifically development of neutralizing antibodies. And so you can see, just as I showed you in that sort of typical graph, you see this spike up through 3 weeks. But what's striking here in both cases for both total antibodies against coronavirus and then the neutralizing antibodies is that by the end of 1 year, they waned. And so there's evidence that we have a much more rapid waning of antibodies to coronaviruses in this case, and I'll show you more with the other coronaviruses. But an encouraging note, and again, this is a different family of coronaviruses, but when they took those patients, the study participants, and looked at them a year later, reinfected them with the same coronavirus they infected them at the beginning, 6 out of 9 became reinfected. That's the bad news, but none of them developed a cold. So it could be, and again, this is just one study, but it does suggest that those neutralizing antibodies might prevent development of disease, even if they don't protect against reinfection. So this is another study, and there's been several with SARS-CoV-1 and the epidemic that occurred in 2003 around the world. And in this case, what they're looking at this is at 3 years, and they're looking at the percent of the population that was positive, the study participants positive for the antibody, and then this is the antibody titer over time. And then over on the right panel, this is just showing a neutralizing antibody titer again as well. And the bottom line is that you could see that by the end of 3 years, a percentage of the people had lost the level of being seropositive, so having evidence of any antibody to the virus, and that would be antibodies coming from those long-lived plasma cells. But in addition, they're losing their neutralizing antibody titers. The same group, Wu Chen Cao and colleagues, also then did in that same follow-up study that was published in 2011. In this case, they took 23 patients that were confirmed for SARS-CoV positive by serology at diagnosis. At 6 years, 21 of 23 had completely undetectable levels. And this is just anti-SARS IgG antibody titers here on the y-axis and months after onset of disease. And this is the threshold of sort of detection. And you can see they're sort of below that threshold of considered to be seropositive anymore. There were 2 patients that maintained antibody titers, and they were trying to understand what was going on with that. But it's clear then that with SARS-CoV, and at least in this study, and there's a few others, that we lose the antibody levels, both total and neutralizing. But the thing about this study, they went and also then wanted to look at memory B cell responses. And what they did is there's an assay which is called an ELISpot assay. And you can take the memory B cells out of the peripheral blood. They looked at them at 6 years later. This is for 23 of the contexts. And you can do as a positive control, you can look at the production of antibodies. And this is patients with SARS, close contacts, and healthy controls. And then they stimulate them with SARS-CoV antigen, non-stimulated or medium alone. And what you can see here, and this was true for all the patients, is a big, especially right here, no response. So they'd lost their memory B cell response to the SARS-CoV. So it was not detectable. So their antibody responses are declining, and the memory B cells, so the two arms, our long-lived plasma cells and the memory B cells, were not there. So that's important to know. And it's shown here in the panel B, just as a control, is to show that they actually had fine memory B cell responses. Total memory B cell responses were fine in the SARS patients compared to close contacts and healthy controls. So this loss of SARS-specific memory was not something in those patients overall to all antigens. It's unique to their response to SARS-CoV. This is another study that was published in Emerging Infectious Disease. And just the key point here is, again, this loss of, this is percent of IgG positive, showing the decline over time, and this is through 3 years, and then also the decline in antibody levels. To look at another member of the family, MERS-CoV. This was a study that was published in 2017 in Emerging Infectious Diseases, looking at patients in South Korea and the MERS response there, and following them out through 1 year. What they show here, and in red are the patients that had a severe disease, and the green were with the non-severe disease, and up here we're looking just at the focus on the neutralizing antibody titers. You can see they're also declining over that year time period in those patients with a severe disease. In the non-severe disease, the level is actually fairly flat and low. And this raises a point that's been question, is whether or not severity of disease contributes to development of neutralizing antibodies, and how that's going to work out for SARS-CoV-2. In addition, this is a sort of a review and JCI. I wanted to bring up the point here, is that we see both in SARS, MERS, and SARS-CoV-2 that we see lymphocytopenia. And we see a decrease in the CD4 positive T cells. And the question I told you, as you remember, to get good memory B cell responses, you need follicular helper T cells. And so an important question that we're going to need to address is there a failure of those helper T cells to work with the memory B cells to develop that long-lasting memory B cell responses? And again, those are questions we can't answer, yes, because we're too early in the pandemic, but we need to keep those in mind. In addition, when we think about antibodies, so that was sort of development of immunity, there is an idea that's been around. It's an old idea for a new pandemic. And the idea is using convalescent plasma from people that have recovered, and whether or not that can be prophylactically given to patients. And we've done this. This has been around for a long time. People do this for rabies, hepatitis A, poliovirus. So post-exposure prophylaxis is a well-known therapeutic treatment. The question is can we use this passive antibody therapy for transfusion of convalescent plasma? It was used in past SARS, MERS, and the Ebola epidemics. The numbers weren't high. The question we have to ask is doing appropriate clinical trials, and actually UC Hospital is a lead site in the Denver metro area for working on developing some of these clinical trials, are participating in clinical trials to look at the use of convalescent plasma. So we'll keep that in mind and see where that takes us. And then the last thing I just want to say here to wrap up is to think about, there's an idea that's been out, and I just feel like if we're going to talk about antibody-mediated immunity, this concept from an epidemiologic point of view called herd immunity or community immunity. And the idea is herd immunity is where a sufficient portion of the population is immune to the infectious disease through either vaccination, which is the key point, and or prior illness, to make it spread from person to person unlikely. And shown here is just a diagrammatic representation. So if you have in red the one infected individual and everybody is susceptible, you're going to get a high proportion of people being, nobody's protected. As the infection proceeds within a community, you're going to get people that have recovered and therefore are thought to be immune. And they have indirect protection against these susceptibles over here just because of the dynamics of that infection. However, if you do use the math models, what you can see is that it's estimated at least, and probably would be greater than as we begin to get a better understanding of the R naught, at least 70% of the population would need to be infected and immune to SARS-CoV-2 to have herd immunity from natural infection. So that is not really a viable or feasible or good option to have as a way to contain this pandemic. So it really is the development of the vaccine to create this population for the protection of the population. So I'm going to close there with some key questions that I've been thinking about. They're certainly not exhaustive or exclusive. And first, is there cross-reactive immunity to circulating coronaviruses? We know we've had these seasonal coronaviruses. This question comes out. There could be some cross-reactive immunity depending on the antigen, but not all antigens are the same, and the SARS-CoV-2 has some unique antigens, but we need to really understand that better. What's the duration of immunity? As I said, we've got data to suggest there's a waning of neutralizing antibodies and loss of memory B cell response to SARS-CoV-1, and is that true for this as well? Is reinfection possible? And if so, does that enhance or limit disease? There's some suggestion there could be some enhancement of disease. We hope it's a little more on the side of limitation of disease, but we don't know anything about that. There is some data coming out of Korea and China that there is reinfection going on, but we'll have to wait to hear more about that. Does disease severity correlate with development of antibody responses? Again, some people with very severe disease might make better immune responses. And all of those questions we just don't have an answer to as yet. And then I think as a lead off to our next speakers, what are the prospects for vaccination? So I'm going to stop there and pass it off to Ross Kedl.
Rosemary, thank you very much. Ross, are you all set?
Yep.
Good.
Okay, good. You guys can hear me fine?
Yes,
Okay.
Perfect, thanks.
Okay, good, there. Thanks, Rosemary. So I'm just gonna cover sort of basic vaccinology, the various platforms that we have used and are currently in use, and hopefully introduce the variety that are out there as a forerunner to what Florian has to talk about specific to SARS-CoV-2. So this is sort of the schematic of the immune response. This is essentially why we vaccinate, because the primary response takes a longer time and it tends to have a lower magnitude to develop. But once it has developed and you have the capacity for a secondary response, it is sort of a classic Olympian higher, faster, stronger kind of a response. It has an increased frequency. It has a much shorter duration to expansion. It's much more sensitive to inflammation, and one of the, much like real estate, it's location, location, location. A lot of the immune cells are actually within the tissue ready to respond. So whereas the primary response, the innate drives the adaptive, actually it's been shown in the secondary response, the adaptive actually drives the innate, which gives it much greater specificity and considerable less collateral damage that the innate immune system is known for. So I'm going to do a very quick overview of kind of immune response start to finish in just a few slides here, and that'll become relevant when I talk about the different platforms of vaccination, because each vaccine platform engages different levels of what a natural immune response would elicit. So can you guys see my pointer here, or does this?
Yes, it shows up.
It does, great. So upon initial infection, this is my representation of SARS-CoV-2. There are innate cells that are drawn into the tissue. There's a tremendous amount of inflammation that's happening within the infected tissue site due to the tissue damage, as well as the triggering of a host of innate receptors. That triggers a process by which antigen-presenting cells within that tissue acquire antigen, migrate to the local node, and begin presenting them to the CD4, CD8 T cells, commensurate with antigen as well as dendritic cell broad-in antigen stimulating, whole antigen stimulating the B cells. This is a process that takes somewhere between, it initiates within hours and it takes within a few days. When things really ramp up, somewhere in the 3 to 10 days range, you have a tremendous expansion of both CD4 and CD8 T cells. The CD4 T cells can enter the B cell zone and begin a collaboration with the B cells and provide help, which induces short and long-lived plasma cells to plasmablasts to produce a ton of antibody, as well as eventually become long-term plasma cells in the bone marrow. The CD8 and CD4 T cells rush into the periphery and now have, whereas previously they had no access to the peripheral tissues, activated T cells can now get in and begin the process of eliminating the pathogen. And as my graduate school advisor always used to say, at the end of all this, an immune response and a Shakespearean tragedy have a lot in common. Pretty much everybody's dead. By the time the virus is eliminated or the infection is eliminated, most of the 95% of the T cells have died off. But again, as I mentioned, there's a key difference. One, the precursor frequency is higher. Two, their sensitivity to inflammation has dramatically increased. And three, their location is now within the tissues. I believe I concluded with the conclusion of the immune response in which the memory pool is now both CD4, CD8, and B cell responses. In response to a full bore infection, is set and ready to go to provide rapid protection upon reinfection. So we have three major categories of vaccines that we do: a live attenuated, in which the infection still proceeds. There is an actual infectious process, but it is virulence is restricted so that it provides immunity without pathogen caused virulence. We can take that infectious agent and just kill it and fix it and leave it intact and then use that as a vaccine platform. Or we can take bits and pieces of it that will serve as the targets of the immune response and incorporate those into an adjuvant, which we'll discuss in a minute. So if we do the live attenuated, the advantage of this is that it captures all of the things I just discussed in terms of how an immune response goes from beginning to end. It's a true infectious process, and as a result, the magnitude of immunity is extremely robust, and it tends to be considerably more durable than other platforms of vaccination. So we get both cellular and humoral immunity, very robust CD8 immunity, in fact, and that's a huge advantage, because one of the reasons why, even in the face of waning neutralizing antibodies, SARS-CoV-2. Memory to that, the T cell memory might be what is sufficient to promote, even upon reinfection, keep somebody from being sick. And there's some good evidence for that in other viral infections. The full engagement of the CD4 response also gives you a much more longer lasting humoral response with a higher titer and better affinity. The disadvantages, of course, are it's an actual infection. You get a significantly higher risk of adverse events and reversion of virulence. There are increased contraindications. It's harder to produce and maintain. And of course, not everything can be attenuated. And there are many examples of that: HIV, so far tuberculosis, etc. So some examples of the vaccines we continue to use and have used in the past are down below. So the next kind of vaccine is really the whole killed vaccine. And this is where you take the infectious agent, you have to grow it up, and then you fix it by formalin or some other chemical means, and then you utilize that as the vaccine platform. Now, the advantage of that is you maintain an awful lot, as this sort of indicates. You capture a tremendous amount of the inflammation that happens at the injection site, because all of the varied innate, other than replication, all of the innate stimuli and innate triggers for all the different innate pathways are still present within that microorganism. You tend to lose your CD8 T cell responses for a variety of reasons we won't go into, but you still get very robust CD4 and B cell responses, and typically a very strong production of antibodies. Unfortunately, you also have a significantly higher incidence of sort of local reactogenicity, which is sort of a local injection site problem. This is some data comparing the whole cell pertussis versus the acellular pertussis vaccine, and you can see that there are significantly greater degrees of reactions when you have the whole cell, because of all the various innate stimuli that are in there and the inflammation that it causes. I think I'm very entertained by this particular one, fretfulness. I'm curious as to the metric for that. How does one measure fretfulness? As well as the high-pitched unusual cry, as to distinguish it from the usual high-pitched cry. I'm not sure how they did that, but there's clearly much more reaction to an entire organism than there is to to just components of that subunit vaccine. So that's one of the drawbacks to this entire process. But the immunity engaged is typically stronger than subunit vaccines. And again, we have some examples here. So lastly, a subunit vaccine is, as the name implies, you just take subunits of the infectious agent. Utilize those, you strip away all the rest of the components of the infection, and you target very specific proteins. This has the advantage of being much less reactogenic, but it also really loses much of your cellular immunity. We typically have to infer CD4 T cell responses simply because we get class switched antibody responses. They tend to be very hard to detect by our typical means. So we have a much reduced humoral response and essentially an absent, for the most part, an absent cellular response. Now, the advantages, of course, is that you can you have fewer side effects in this process. You have a much lower production cost and increased shelf life. But this also does tend to require significantly increased boosting, as can be highlighted here if you compare the measles response, where a calculated half-life decay for the antibody is over 3,000 years, whereas you can reasonably calculate a legit antibody half-life for the tetanus vaccine, which is one of the reasons why boosting every 10 years is required. So besides the boosting, this requires the use of an adjuvant. Because the antigen itself doesn't induce any inflammation alone. Being a foreign antigen is not sufficient to induce immunity, as it once was thought in the self-non-self discriminatory world. If that were the case, human reproduction would be impossible, as every woman would mount an immune response against human chorionic gonadotropin, as far as they are concerned, a foreign antigen to them, and the entire life process would stop there. So foreignness is not sufficient. There needs to be some sort of inflammatory component to the entire thing. And so we've developed a cadre of adjuvants to provide the innate immune stimulus that hopefully triggers the entire process. Now, these tend to be very good at inducing, to varying degrees of inflammation. Everyone's probably very familiar with alum, which was developed in the 1920s, and it's essentially the backbone of all of our adjuvants today. There are other various emulsions or precipitates or sort of obnoxious, indigestible components that tend to irritate the immune system locally well enough. They're sort of local irritants that seem to operate outside of standard innate receptors, although the inflammasome is clearly engaged, whereas others that trigger very particular innate pathways or combinations of all of these things. And there's examples of those adjuvants being used very successfully in various sides of the clinic and against infectious agents. But again, these as of yet we have yet to develop adjuvants that make clinically relevant cellular immunity. So there are some newer formulations and adjuvants that are under development, given the desirability of the subunit platform, broadly speaking, for a population. A huge category is this area of nanoparticle formulations. There's a variety of kinds. There's some examples listed here. ISCOM stands for immunostimulatory complexes that are made of typically a mixture of lipids as well as polymers that then form some sort of microparticle-like formation. We can conjugate the known agonist specifically to the antigen of interest, and there's some good examples of those. And then lastly, I'll finish up with nucleic acid-based vaccines that are now being sort of prominently featured and are important in what's going on for SARS-CoV-2. So here's a couple examples of microparticle formulations. Here are liposomes, and these are PLGA, PLA particles into which you can embed or impregnate with the antigen of interest, as well as even other adjuvants. This is useful as it has multimeric antigen for direct B cell stimulation. It greatly facilitates phagocytosis, so the particle is very well taken up by antigen presenting cells. And that actually also helps because of the nature of the chemistry of this. It tends to facilitate the trafficking of the antigens from the endosome into the cytosol. And that's a very important thing if you want to generate CD8 T cell responses. So in animal models, these tend to do much better at generating cellular immunity, although that has yet to be fully realized in primates and higher primates. Anyway, there are a broad range of microparticles that are being utilized, as well as I mentioned before, this idea of conjugation of a TLR agonist to an antigen of interest. This seems to be very useful in generating immunity because it ensures the co-delivery of both the antigen and the adjuvant to the same APC or the same B cell. And similar to the microparticle, it enhances uptake and activation of the APC for cellular responses. Here's a couple examples of those from Bob Cedar at the VRC at the NIH, using sort of a multimerized TLR7 antigen conjugate platform. And then you can sort of group these together, create a microparticle, and then again impregnate that with TLR agonists as well as antigens of interest. So this is kind of using all those different platforms together. Lastly, nucleic acid-based vaccines are sort of on the rise in popularity, in part because they're very easy to produce. Obviously, it's not that hard to make DNA. Once it's made, since we can pull it out of a Cro-Magnon skull, it tends to last a long time, even in the absence of a cold chain of any kind. So it's very stable over long terms and long distances, and if it could be utilized effectively, then we could make vaccines much more cheaply and readily available. So it turns out that if you inject DNA, it can actually get taken up, particularly by muscle cells, and produce the protein that's encoded. By other cells will do this as well. Electroporation is a new process that has been utilized. The previous version of this, and it's still utilized, is a sort of a gene gun technique, ballistically delivered. It has gold particles that are needed, so those get blasted into the skin. Turns out it's not a great idea to base your vaccine delivery method on the price of gold. It makes it a little bit more tricky to price effectively. So electroporation is a new way of doing this, where there's devices that can inject the material, the DNA, into various depths, and then provide a charge across that, and that seems to help and get the DNA as well as RNA into cells of interest and get them expressing their antigen. Good efficacy in animal models, less in clinical. They tend to be most effective if you do a heterologous prime boost. So if you prime with the DNA vaccine and then boost with more common adenovirus or another platform. One of the problems, of course, is that if you deliver DNA, it produces the antigen, and the DNA itself is a trigger for innate receptors. Since DNA in the wrong place triggers a variety of receptors. By the time it's producing the antigen of interest, an awful lot of the time the adjuvant effect has waned a bit. So now you're making a foreign protein without a whole lot of inflammation. So that's a bit problematic with maintaining immunity. So if you actually make the DNA encode an adjuvant along with the antigen, that tends to increase the responses. So if you utilize flagellin or some other protein-based TLR trigger, that tends to be very useful. And lastly, mRNA is now what companies are already off the ground and using in the clinic for SARS-CoV-2. In vitro transcribed RNA has been packaged in a whole host of ways. Obviously, RNases are a problem, and the degradation of it is a problem. So a variety of methods have been used to package it and deliver it with nanoparticles or polymers of one kind or another, or to alter the sequences to make it less digestible. So a whole host of adaptations of RNA are used, and obviously its capacity for rapid production and delivery in a variety of ways makes it a very significant platform of interest. It has similar problems to what's been seen with our DNA thus far, and time will tell as to whether this will be a useful platform or not. And I think Florian has a few things to say about that. So challenges for SARS-CoV-2, really an optimized vaccine platform. There's 3 that I know of currently in the clinic, although I think Florian will update us on that. We need some metrics for comparing them right out of the gate, so we have a good idea which ones to proceed most effectively with. Adjuvants always are in sort of need of optimization. Obviously, we need neutralizing antibody as the primary objective. If there was a possibility for cellular responses, that would provide greater cross-protection against future pandemics, since a lot of the internal antigens that cellular responses are driven by tend to be a little bit more consistent over time in terms of their sequence. And then if we provide this prior vaccination, as Rose highlighted, there was a waning of antibody responses over time to overt infection. Will this help prevent the loss of that after vaccination and subsequent infection? And then more broadly, the idea of formulation and manufacturing, there's a global demand here. So very small differences when you're trying to vaccinate, 7 billion people? Small differences in vaccination costs can really make a difference. So, and the distribution, again, global demand. There's a whole bunch of struggles with that. So those are all going to definitely come into play as we consider what the best platform is in providing protection. Okay, so that's it for me. Florian, I think you have the floor.
Ross, thanks. Sorry for the IT problem.
Yeah, my bad. Sorry about that. Let me unshare here.
Florian.
All right. Thanks a lot. Trying to share my screen here.
Thank you for joining us.
Thanks for inviting me. So what I'll try to do is I have a few slides about the immune response to SARS-CoV-2.
You may want to put this in the present. Good, great.
Okay. Need a pointer. So what I'm going to do is I'm going to share a few slides about immune response, specifically antibody response to SARS-CoV-2 that we started to study here at Mount Sinai, and then I'm going to give a more global overview of vaccine candidates and vaccine design and production for SARS-CoV-2. So let me just start, and Rosemary was talking about that already, about the virus itself. We have a model here on the left side. You can see it's an envelope virus, so these yellow lipids here, that's the host-derived envelope. We have a major glycoprotein on the surface, and that's called the spike protein. And then we have a matrix protein in green, and an envelope protein. And on the inside, we have the genome that's covered by the nucleoprotein.
Florian, could you just get a little closer to your microphone? I think it just keeps fading in and out.
Yeah, that microphone is actually directly next to my, I have a headset on, so that should work. You can try to do it without the headset. Let me see if this is better. Can you hear me better?
Yes, it's good.
Okay. So we are focusing mostly on the spike protein here because we know from work with other coronaviruses that the neutralizing antibodies that you get against these viruses are actually directed against the spike protein. And so the spike is, in terms of humoral immune response, the most interesting, and it's also very interesting for vaccine design. And what I'm showing you here is the spike protein. It's a trimer. So I have one of the monomers here in dark blue. The other two monomers are in light blue. And then what I show here in red is one of the receptor binding domains. So that's the part of the virus or of the spike that engages with human receptors. And that's actually where most of the neutralizing antibodies bind. And so we started early on to develop serological assays and serological tests to measure antibody responses against SARS-CoV-2. My lab is usually doing this for influenza and hantaviruses and arenaviruses, and so it was kind of natural to do that for the coronavirus too. And we started to work on that on January 10th when the sequence was released. And so why are we interested in serological tests here? First of all, Rosemary pointed it out very clearly. We need to understand the dynamics of the antibody response to the virus, and you can use these tests for doing that. There's also a lot of interest in serosurveys to find out the true infection rate. And what we did early on here at Mount Sinai is we looked for people who had already high titers post-infection, and we asked them to donate convalescent plasma so that we could treat other severe cases. And if you have an assay to actually test how strong an antibody response is in a donor, you can select the donors with the strongest antibody responses for plasma therapy. And there was also an interest, and still is, in identifying healthcare workers that are already immune because they had mild or asymptomatic infections. And as I said, we started relatively early on with that. What you need in order to perform these assays are the right reagents. And so we made two versions of the spike protein recombinantly. And I'm showing you here the constructs. One is basically the full-length version of the spike protein, just without the transmembrane domain, and with a few stabilizing mutations. So this forms a very nice soluble spike. And then we also looked at this receptor binding domain only. And so in January and February, we started to make these proteins recombinantly, both in insect cells and in mammalian cells. You can see these proteins here in their purified versions on STS pages. So it's relatively easy to make them. The spike is relatively large. It's more than 140 kilodaltons large, which makes it harder to express. The receptor binding domain is relatively small, and that makes it easy to express. And so we started to set up assays here with sera from people who we had recruited before the SARS-CoV-2 pandemic, and then also patients from Mount Sinai, but also initially from Finland and from Australia. And I'm just showing you some results here, and this is reactivity against the receptor binding domain and the spike, the full-length spike protein. In black, you have 50 controls. One of the controls is actually shown in green. That is a control serum taken 30 days after NL63 infection. So that's also a human coronavirus. And the red ones are all patients that were infected. Now we have a lot of data. We have tons of additional controls. We don't see cross-reactivity from human coronaviruses in that assay. And what I also wanted to point out is, so there's different types of samples in here. There are samples from people who have been hospitalized. There are samples from mild cases, and there are samples from asymptomatic cases. We find reactivity in most of the people that tested PCR positive, but there are differences, and people who are severely sick usually make more and stronger antibody responses, while in asymptomatic and mild cases, we see a later onset of antibody responses, and the antibody response is usually a little bit weaker. So we're studying now how long-lived these antibodies are, and that's something we can't answer yet. What we did is we also performed correlation analysis between what we measure in these binding assays and what we measure in neutralization assays. And so for many viruses, neutralizing titers are correlated with protection. But for this virus, neutralization assays are hard to do because you have to do them in a biosafety level 3 laboratory. And so if you can establish a correlation with another assay that is easier to do that, that's worth a lot. And so we see that we have actually a very good correlation between binding in this ELISA and neutralization. What we are doing now is we are setting studies up to correlate both neutralization titers and ELISA titers with protection from reinfection. This will take a while, at least a few months, but that's something that we are currently conducting. And so just in conclusion from the serology studies that we have performed, we see that the general population is completely naive to the spike of SARS-CoV-2. And that might explain the rapid spread of the virus, because there seems to be no pre-existing titers against or neutralizing titers against the surface glycoprotein. And this also questions if antibody-dependent enhancement could explain pathogenicity in people who are older than 50, where we see most disease. There was a hypothesis in the beginning that older people might have more cross-reactive antibodies, and that might drive pathogenicity. But we didn't see a difference in younger versus older individuals in terms of cross-reactivity. There was just none. We also see that humans induce a solid antibody response to SARS coronavirus 2. The question is just how long-lived that is. And my lab is a research group, but we transferred our ELISA setup in March, beginning of March, to our clinical lab. And they last week got FDA emergency use authorization to use that and diagnose if somebody is, as a diagnostic to find out if somebody has been infected with the virus or not. Not for acute cases, but to basically find out if somebody's seroconverted. And we have also used this extensively to find plasma donors. We have dosed more than 100 people at Mount Sinai with convalescent plasma. The open questions here are how long does antibody-based protection last, and what is really the protective level of antibodies that we need. And that leads me to the second part of the talk, which is about vaccine development. And I'm just showing you an overview here. Again, on the left side, you have the model of the virus. We know from other coronaviruses, as I said earlier, that the main target of neutralizing antibodies is the spike, and specifically the receptor binding domain. And so that's what vaccine development currently is really focusing on. There's a number of different platforms that are in use. And then there's a whole process of how to get these vaccines onto the market. And that's what I will be discussing in the next few minutes. But let's start with the vaccine platform. So Ross already presented a lot on different vaccine platforms and their advantages and disadvantages. I just wanted to show what's in development right now for SARS coronavirus 2. There's more than 80 candidates in development right now. Most of them are in the preclinical stage, meaning that they are getting tested in animals, but they're not in humans yet. We have RNA vaccines, and they can come as messenger RNA or replicon-based vaccines. This is a very new concept. There are no there's no RNA vaccines on the market right now. They seem to be easy to scale, and the ones that are in development target the S protein. DNA vaccines are also not on the market. They are in development now for a long time. There were potency issues initially. They work well in mice, but haven't worked out well in humans. But we also have now DNA vaccines in development against SARS-CoV-2, and they also target the S protein. Again, DNA vaccines are probably very safe, and they're easy to manufacture. Then there's recombinant protein vaccines, which again target the S protein. And we have recombinant protein vaccines on the market. There's one for influenza, Flublok, and then there's HPV and HPV vaccines that are also on the market. So there are companies out there that know how to make these vaccines in larger quantities, and that's an advantage. And there's a kind of a known route to licensure, which is also an advantage, because if you come with a very new vaccine, very often regulatory agencies are not very familiar with those platforms, and it takes much longer to get these vaccines to market. We have virus or vectored vaccines, and so far there's only one that is licensed based on a VSV vector, and that was licensed in the European Union by EMA, which is the European counterpart of the FDA. And there's now many virus vectors that are getting developed as vaccines for SARS-CoV-2. And again, they target all the spike protein on the surface. There is a lot of safety data with these vaccines because they have been in clinical studies for many different diseases for quite some time. And so that's certainly an advantage. We have inactivated vaccines where the virus is just getting basically chemically or physically inactivated. This is a very old technology, but it's also straightforward. And there's still a lot of vaccines on the market that are developed, that are produced that way. So there's probably a lot of production capacity globally to make a vaccine like that. And then we have live attenuated vaccines, which are also in development for SARS-CoV-2. The issue here is a little bit that the virus is a biosafety level 3 virus, and it has a huge genome. It's not so easy to manipulate. So if you make, by reverse genetics, live attenuated vaccine, you have to get it out of biosafety level 3. You have to basically show that it's safe first. And so for safety reasons, making live attenuated vaccines against these viruses is not so easy. what I also wanted to discuss is the timeline and the issues that that these vaccines have to get through before they can get to the market. So everybody's asking, why does it take so long to make a vaccine? Why don't we have a vaccine right now? So typically it takes about 10 to 15 years to bring a vaccine from the laboratory to the market. And what is projected now for a lot of these vaccines is that they will be on the market in between 12 and 18 months. Some are even more optimistic and say 6 months. But there's a lot of things that need to happen to get them to the market. First, you need to have an appropriate process to make the vaccine for use in humans. That's called GMP, or good manufacturing process. And so for some of these vaccine platforms, pretty good processes exist. For others, they don't really exist, at least not at the extent needed. And so there's a lot of development needed that goes in parallel to actually develop the vaccine candidate. The vaccines, once they have been produced, need to be tested in clinical trials. That includes a phase 1 trial, which mostly looks at safety, a phase 2 trial, which looks at immunogenicity, formulation, different doses, and then you typically have a phase 3 trial, which looks at effectiveness or efficacy. This usually takes years, and now companies, but also regulatory agencies, are trying to compress that so that it can be done in a very short amount of time. What needs to be kept in mind here is that we cannot compromise safety. And so safety evaluation in humans is really, really important. And that's also why these trials have to be done thoroughly, and why it takes quite a while to complete them. Once the clinical trials are done, basically FDA, EMA, other regulatory agencies have to license the vaccine so it can enter the market. But another problem that Ross mentioned is we need large-scale production because we potentially have to have maybe 7, 8, maybe even double that amount of billion doses, right? So basically we need enough vaccine to vaccinate the global population. And it's very likely that a lot of these vaccines have to be given as a prime boost regimen, meaning that they have to be given twice. And so again, for some of these vaccine platforms, we have large production capacity. For others, we don't. We don't know how to make RNA vaccines or DNA vaccines at the scale needed. The facilities just don't exist right now. And then the vaccine needs to be distributed. And that can also be an issue because in many countries where the vaccine would be needed, there are no cold chains, or it's really hard to distribute and get people vaccinated. And so that needs to be kept in mind. And once the vaccine is administered, we probably have to wait maybe even like 3 weeks after the second vaccination until people develop immunity. And so this is why it takes quite some time to bring these vaccines to market. The positive side here is that we have some experience with coronavirus vaccines, not extensively, but with SARS coronavirus 1 in 2003, there were many vaccine candidates, and 2 of them actually made it to clinical trials. Those were early stage phase 1 trials, but it's still important to look at the data. One of the trials was with a DNA vaccine, and that was performed at the Vaccine Research Center at NIH. And it was just in a very small number of individuals. And some of these people made actually relatively high neutralizing antibody titers, but some of these individuals didn't make antibody titers at all. If you look at A here, for example, or B, so it was very heterogeneous. There was a second trial that was performed by a company in China, Sinovac. And what they did is they just made an old school inactivated vaccine for the old SARS coronavirus. And they gave 2 vaccinations with that vaccine, and they got pretty nice neutralizing antibody titers too. And so that's the experience that we have. There were also a lot of preclinical experiments with different vaccine candidates. In some cases, there were complications when vaccinated animals were challenged. So it's not that straightforward. And there can be, I wouldn't say enhanced disease after vaccination, but there can be complications. And that makes it even more important to really look at safety. And that's where we stand right now. As I said, there are approximately 80 candidates that are being developed. And of these 80, so far, 6 have made it to clinical trial, or 5 have made it to clinical trials. One is on hold, it seems. We have one candidate from the University of Oxford, from the Jenner Institute, and that's based on an adenovirus vector that expresses the spike protein. So this clinical trial has started and is in phase 1. A Chinese company, CanSino, has another adenovector that they're developing. It's an adeno5 vector. I think they're about to concluding their phase 1 trial, and they're starting a phase 2 trial now. The company that I just mentioned, Sinovac, that made this inactivated vaccine against the initial or the classic SARS virus from 2003, has also made an inactivated virus vaccine, and they started a phase 1/2 trial. I'm not sure about this bifidobacterium vector here. That trial seems to be on hold. The Vaccine Research Center at NIH, together with Moderna, has moved a messenger RNA vaccine forward. And so this messenger RNA vaccine is also coding for the spike protein. And that was actually the first clinical trial that had started now a few weeks ago. And I think the initial trial site was Seattle. But they expanded that now to 2 more trial sites. And then the last candidate is a DNA vaccine, again focusing on the spike protein, that's developed by Inovio, a company in the US, and they are currently conducting a phase 1 trial as well. The interesting thing here is that a lot of these companies moved into clinical trials without publishing much of their preclinical data. The Moderna VRC vaccine didn't have preclinical data so far. They based their data package for the FDA on other vaccine candidates that they had in the same platform. So it's going to be very interesting to see preclinical data and to see how these vaccines perform in animal models that are then challenged with SARS coronavirus too. And so for one of these candidates, last week, preclinical data came out, and it's actually pretty good data. And that's the inactivated vaccine by Sinovac. I'm not going to walk you through the data here. Unfortunately, the preprint that they put online had low quality figures. It's kind of hard to read this. And they also performed very thorough safety studies, and they published that data online as well. This here is data from non-human primates, and what they show is that when they vaccinate in a prime boost regimen with this inactivated vaccine, they get good, relatively solid titers of neutralizing antibodies, and the animals are protected from challenge with SARS coronavirus too. So this is a non-human primate model. I think it was a rhesus macaque model, which is a pretty okay model for SARS coronavirus too. So this is very positive data. So to conclude on the vaccines, vaccines are being made in record time. I think it's the first time that we see vaccine development happening in that short time frame. However, it will take very likely until 2021 for the vaccines to be available. It might be that China has a vaccine a little bit earlier, just because they might have more production capacity and the regulatory agencies might be less strict. The important point here is that safety needs to be assured. There were complications with vaccines against the old coronavirus in some animal models, and that needs to be investigated before we go and vaccinate millions or billions of people. And what I also wanted to say is that more than one vaccine will be needed. There's not one single company that can satisfy the market right now, and it's very unlikely that just one company might be able to deliver enough doses, even over time. So we need many vaccine candidates and likely globally distributed production facilities. And the last point that I don't have on this slide is many vaccines that we use don't work very well in older people. And that's the most important group now for SARS coronavirus 2, because that's where the mortality is the highest. So we might run into issues in that age group. And I think that's something that needs further investigation to see if we get good antibody titers and good protection in people above the age of 70, 60 or 70 as well. I think that's something that people have to keep in mind. And that's it for me. And moving back to the slides from the beginning.
Florian, thank you very much. Thanks, Ross and Rosemary. We're going to now go to the open question session. Let me moderate the questions because I have them in front of me, and I'll ask all the speakers to just respond if they want to these questions. The first has to do with herd immunity. So how does herd immunity relate to all of this in terms of the number of individuals or the percent of individuals that are needed to become immune, either by virtue of previously being infected or by virtue of being vaccinated to SARS-CoV-2? And how do you calculate that in a population-based way? What are the variables that go into that calculation?
Since I was the one that brought it up, I don't actually know all the variables that go into that calculation, so I have to confess that I'm not an epidemiologist by training. However, you do have to know the proportion of people that are infected within a community already. You do have to know the R0, the transmission potential of the virus. And when we think about that calculation, I could just say we know that measles virus, which is one of the most, has the highest R0 of any of the viruses, is of an R0 of 18. So one infected person can infect 18 others. You have to have 95% of the population vaccinated or to protect and generate that herd immunity. So vaccination is really especially with a pathogen like SARS-CoV-2 or even with measles virus that causes significant mortality. You need to have a vaccine rather than having any kind of hoping that we'll all get immune at some point.
And Florian, given your studies with antibodies, what is your estimate on the background rate of infection in the general population?
Well, that will differ from location to location in the US, right? I think the estimate here in New York right now is about 6 to 8%. And we're starting to see numbers in seroservice that basically are coming close to that number. So I don't think we're anywhere close to herd immunity anywhere in the US. And there's already a lot of dead people. So I think to reach herd immunity would cost a lot of lives, and I don't think it's worth it.
So related to this, this antibody response, the question is the somewhat dissonance between symptoms and antibodies. In other words, individuals that are more symptomatic seem to have more of a humoral response. Is that a cause and effect relationship? Are individuals symptomatic because of the substantial immune response? And are individuals asymptomatic because of the lack of an immune response? Why do asymptomatic individuals seem to have minimal effects of this virus, but don't have a profound immune response, at least via humoral immunity?
I'll let Florian answer that one.
I can try. I think if somebody is severely sick, there's a lot of innate immune responses that happen. There's a lot of antigen floating around for a very long time. And so you engage a lot of CD4 cells, you get probably a lot of germinal center reactions. You just get more stimulation, right? In somebody who gets an asymptomatic infection, that might happen to a certain degree, but probably not to a very, not very strongly. And you're completely right. That might correlate with the symptoms, right? If you don't get interferon or a strong interferon response, you might not feel sick. So I think there is certainly a correlation there. The question really is, in the end, what is a protective antibody titer? We see that the antibodies come up in people who are asymptomatic too, and maybe you don't need a titer in the 10,000s in order to be protected. Maybe a titer in the hundreds is enough. So that's something that we need to figure out.
Ross, why don't you unmute yourself so that you can answer these questions as they come up also.
Yeah, I'm sort of compelled by the data on IL-6. I mean, IL-6 is definitely a driver of TFH and germinal center reactions. And so the idea of that rising IL-6 tracks with substantial sort of immune-related, inflammation-related pathology, I think that tracks nicely in my mind with the rising antibody response that isn't causative, but more of the canary in the coal mine that the inflammation is spinning out of control.
So let's talk a little bit about the innate immune response as it relates to antibodies as well as cell-mediated immunity. Does this account for why some individuals like diabetics or the elderly are more susceptible? How can you connect the dots between these susceptible populations and
That's the million-dollar question, though, isn't it? What makes some people more susceptible to
That's why we're asking you.
I guess we're the experts. I think it's really curious that vascular issues seem to track with increased susceptibility, and so that's curious to me. I don't know enough about vascular physiology to make any intelligent statement about that. Not that that's stopped me historically, but that is of interest to me. It's not surprising that overwhelming immunity and a lot of cytokines can be detrimental to the functioning of a normal lung. So that's not surprising. But exactly why something like diabetes is tracking with it, I think that's fascinating. I don't have any great ideas. Anyone else?
Florian, do you have any ideas?
Yeah, I have hypotheses that are completely unproven. So I don't know about comorbidities, but the old versus young picture that we are seeing is not really new, right? People get vaccinated against measles, mumps, so old people don't get measles because they're vaccinated or they had it when they were young, right? But if you would infect children with measles and you would infect 70-year-olds with measles, the children would suffer, but they would survive and they would do fine, most likely. So somebody who is 70 years old and gets a measles infection would likely die. And so I don't think that the pattern is very different from most virus infections. And that might have to do with kids make a lot of interferon quite quickly. And it might be that the virus has a very long incubation time, right? So you get infected, and in cells of younger individuals, you might locally get a relatively strong immune response that shuts down the virus. And it has been shown that SARS coronavirus 2 is actually more susceptible to interferon than SARS coronavirus 1. So it's pretty sensitive to interferon, right? So, and young individuals might just locally make that, and the virus replication might be shut down. In older people or people who have issues with their immune system, the virus might just keep replicating until there's so much virus that the innate immune response is triggered, but to an extent where the innate immune response then becomes the problem, right? And the immune response in general becomes the problem. There are some studies, and there are preprints out about that kind of suggest that in terms of induction of the innate immune response, but then there's no really good evidence that would support that.
Rosemary, you have something to say?
Well, I actually want to echo what Florian's saying in terms of the very young and very old. And there's a with influenza, we see that the very young very susceptible, and then as you get older, you're more susceptible. And we don't have enough data on the extremely young. We know that there are kids that are getting seriously ill with SARS-CoV-2, but we could also think about there's some hypothesis out there, and I don't know if it's all of this we have to get more data in about whether there's something that is co-infection or some other trigger in the elderly population that might exacerbate that response, so that they might have an I call it a PTSD of the immune system. It remembers something that it shouldn't and overreacts. And so we don't know if there's something that's triggering it in the older population that might be of that mechanism. So that's another possibility as well in terms of that. And there's also some suggestion that there's autoantibodies that are made in the more serious infections. So you might have some triggering immune response to SARS-CoV-2 that's reflective of something you've had earlier in your life. And I think all of these are important areas for all of us to investigate.
I mean, one of the things that to me is really striking about the death curve to SARS-CoV-2 is its extreme correlation with age, and that it doesn't have that bimodal peak. That very young are really not affected in the way that they tend to be to viral infections, like almost all the other ones we can think of. And so there really is the most extreme correlation here is with age. So something very significant is happening there. I think it's really interesting, this idea of innate versus adaptive. It's well known that aging, the capacity to generate new immune responses drops off as we age. But one of the things that lymphocytes have always been implicated in that we don't make new lymphocytes. But actually, in the animal studies have been done, if you take very old lymphocytes and put them into a new mouse, they do just fine. They do remarkably well. In fact, in many ways, they do better than young lymphocytes. So what is really one of the things that's come out of that is that the innate immune system is, as we age, that is actually garnering far more inappropriate ways of responding than necessarily the adaptive side of things. And so that very much tracks with potentially what we're seeing in terms of the pathology of this.
So what's unique about this virus that results in a fall off of our immune system in terms of humoral immunity?
I'm going to jump in on.
Or let me just ask the opposite side of this question, which is this so unique among viruses, or are there lots of other viruses that do similar types of things in terms of a fall off of humoral immunity?
I'm going to jump in from a parasite perspective. So I also study malaria, and in malaria, you don't get good development of humoral immunity. It takes multiple rounds of infection to ultimately develop antibody responses. And so what they've discovered is the T follicular helper cell is pretty critical to the development of immunity, and that's messed up during malaria. For other viruses, this one seems to be fairly dramatic in terms of the fall off for the SARS coronaviruses. So, I mean, not the SARS, the beta family coronaviruses. So I study EBV, and antibody responses go up and they stay up, but that's a persistent viral infection. Influenza, we know that the antibody, Florian could speak to that better than I could, but those go up and stay up as well. So I think there's something about how this virus is damaging the development of immunity that is critical, either through an inflammatory response that's skewed in the wrong direction, loss of the T follicular helper cells, if that lymphocytopenia is actually by apoptosis rather than just our inability to track them on migration of the lymphocytes to tissues. So I think those are important questions.
Other responses to that question? No. Let me move on to another question. I'm sure we could talk about this for a while, but the question relates to convalescent serum. Should we assume that convalescent serum is working, and to what extent should it be subjected to a rigorous clinical trial?
I mean, maybe I can start to answer that. And so I think clinical trials are always good. There was a discussion here at Mount Sinai too, but our clinicians felt that if there is a treatment, everybody who can get it should get it. And that was at the point in time when the situation here was really getting out of control. And it's still pretty bad. So I think there is a need for clinical trials. There is clinical trials ongoing in China. As far as I know, there's also clinical trials getting started here in the US. And it's important to look if it's really working, but it's also important to figure out what you're actually doing, right? Because convalescent plasma works in some cases very, very well. There's even a product on the market in Argentina that's made of convalescent plasma against Hunin virus infections. That's a licensed product and it works very well. In other cases, convalescent plasma didn't work that well. I think it always has to do with the potency of the plasma, and what people tend to do is they just like look for people who survived or recovered and then use that without a good characterization of the activity of that plasma. And I think that's kind of the key. And the other point is really, we're dealing with two different problems here. The first problem is the virus, right? You have virus replication that damages the lung. But at a certain point in time, the virus is not that much of a problem anymore. The immune system that attacks the virus becomes the problem. And I think if you're at that stage, the use of convalescent plasma or the use of antivirals is kind of not that helpful anymore, right? So we really need studies to look at how this should be ideally used and where it makes the biggest impact. So that's my opinion.
Why? So in that vein, why aren't we seeing more immunocompromised hosts come in with overwhelming coronaviral infections?
They're not going out.
I hope so. I hope they stay
Well,
on.
hopefully they're not being exposed.
They're not gaining access. I mean, I think the answer to your original two questions are yes and yes. Yes, we should probably be using everything we can, and yes, we should get better testing. I mean, generally speaking, I can't think of an example in which, like the worst case scenario of convalescent serum is it didn't work very well. I'm not aware of, like, I imagine if you did it with dengue, where there's some curiosities with being infected with one virus and then struggling with a different strain, that possibly in that circumstance, you might experience something worse with convalescent serum. I don't know if that's been done or not, but the worst case scenario with convalescent serum tends to be like not much. So the chances of having a benefit seem to outweigh the risks involved in the current circumstances. I think it makes sense to use it as readily as possible. And it would be extremely good to keep track of that as we go along. Who did it come from? I think something that I've been thoughtful about here as we try and maintain the response against COVID-19 is catching as much information as we possibly can as we go through. Who have you been in touch with? Who do you know? What is your serum levels of antibodies like, so that we can find out, and who did you get it from? And being able to track those sorts of things might give us an insight in sort of a meta-analysis kind of a way, without an organized clinical trial, that might give us some meta-analysis to mull over as we recover from this whole thing.
This leads into the next question, which is what are the determinants of the host that would improve their chances of responding to the vaccine? In other words, are older individuals going to be less likely to respond to the vaccine? Are individuals that have risk factors for SARS-CoV-2 infection going to be less likely to respond to the vaccine? Are individuals who responded to other vaccines like influenza going to be more likely to respond to the vaccine?
I can try to address that. It's complex, right? You would assume that if you give a vaccine to somebody who makes typically a good immune response to, let's say, an inactivated vaccine, you would assume that you would get a good immune response here too. It depends a little bit on the vaccine platform. We don't know enough about the messenger RNA vaccines and the DNA vaccines yet to see who are people who respond typically well and who are people who don't respond. But I think will likely follow similar trends as you see with other types of viral vaccines.
Mhm. Okay.
Yeah, to favor your response, potential response to a vaccine, if you can be a young Olympic athlete, then I think that's your best foot forward. Like,
Mhm.
those are the likeliest ones to respond well to a vaccine. Beyond that, that's a tough one to answer.
And I mean, that's why I brought up that point earlier about vaccinating the elderly, who are really the ones who need that vaccine. And we might not get good responses there. But on the other side, if you get a very high vaccination rate in a country and you can build herd immunity, you would get an indirect benefit for people who don't respond well to the vaccine as well.
Yeah, I mean, it brings up the question. They've done some interesting studies in flu vaccination where they, vaccinate everybody below the age of 12, everybody over 65 or both. And you can protect most of the 65 years and older, it turns out, by just making sure everybody under 12 has been vaccinated. So in part because, Johnny brings it to grandpa, and that's part of the problem. But the other part is that, the elderly, it is harder to vaccinate and generate really robust immune responses. And so kids make a much stronger one. They provide a much better barrier. So maybe, brings that question, maybe the first line of defense is hit up everybody below 12 and above 65, and then maybe the middle of the road. I don't know. It sort of leads to some interesting questions about what the best path forward for a broad public vaccination might be.
I'll jump in and just sort of add the other point about Florian had made the comment, we don't know what the correlates of protection are. So when we say what's a good response, right? How much antibody do we actually need to make in terms of being protective, and stability of response, the length. And so all of those are important questions, but we don't, actually I keep on saying for every answer, we don't have the answer to that, but it's a great question, which is true about all of this. But it highlights with this disease, I think it's going to push us like HIV did to really think about pathogenesis in a way that we haven't had to think about because of the nature of the pandemic. All eyes are on it, and all eyes are thinking about some really important questions, immunologic questions, in terms of trying to think about how do we vaccinate a population.
In terms of development of vaccine, tell us a little bit about mutation rate of the virus and whether that's going to present a problem.
It's not flu. All viruses mutate. Even DNA viruses like to mutate, right? So the virus is changing, and that's actually how we track clades and how it's moving around the globe. I wouldn't expect antigenic changes quickly. Some human coronaviruses like OC43 go through some type of antigenic transition, but slowly. Coronaviruses actually have some kind of proofreading activity, so the mutation rates are usually lower than for your typical garden variety RNA virus. And there's another important point that sets it apart from viruses that drift fast like influenza. And that is, influenza has relatively large hemagglutinin head, but the receptor binding site is tiny because the receptor is tiny, it's a sugar. And so basically the virus can do whatever it wants around that binding site and mutate as much as it wants to. Other viruses, for example, measles, bind to a protein receptor. They have a very large binding area. And they can change that easily, because if they change that, they might not bind as well to the receptor anymore. And that's kind of the same game with SARS coronavirus 2. It has a relatively large binding surface with human ACE2. And that's where the neutralizing antibodies bind. And if it starts to change that drastically, the antibodies won't bind anymore, but it also won't bind human ACE2 anymore. And so that might come with a pretty big fitness cost. So I'm relatively positive when it comes to antigenic drift with SARS coronavirus 2.
Mm-hmm. We have time for one more question, and I want to focus on the persistence of this immune response, both in terms of the natural infection and potentially in terms of the development of the vaccine. Given the persistence that we're seeing with the natural infection, will this be a long-lasting response to the vaccine, or will this be a vaccine that we have to administer every several years?
So when we think about the natural infection, there's a lot of things going on during that infection, not just the virus replicating and sort of a very nice immune response, but you've got this inflammation and other things that are going to drive the probably inability to generate that long-lasting immunity. We don't know that quite yet, but based on the other SARS and MERS, possibly. But a vaccine is very different, and we know that vaccines can generate long-lasting immunity. And so it's in a clean system or a clean host where you're not having all these other pathogenic reactions going on that could hamper development immunity. So I think I would be much more optimistic about the vaccine and generating long-lasting immunity than I would about a natural infection.
And I want to add something to that point. We have an advantage here over viruses that are very quick, like influenza. The incubation time for flu is very short, and it hits you hard right away. The incubation time with this virus is relatively long, days, sometimes 2 weeks. So even if your antibody levels go down and you were initially infected, if you have memory B cells, and there are some questions around it, but if you have memory B cells, you can get a very quick plasmablast response. And that might actually take care of the virus infection before you get any symptoms. And I think that's an advantage with viruses like that. So we might be lucky in terms of vaccines, but also natural immunity to some degree.
Well, unfortunately, we've come to the end of our time. This has been a terrific discussion. I want to thank all of you for presenting and sticking around for the discussion period. It's been very valuable. Thanks.
Brilliant.
Inviting us.
Thanks for the invitation. It was fun.
See ya.